Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Our DNA holds an immense amount of information, compactly organized to incredibly tiny dimensions, small enough to fit inside a cell's nucleus. A DNA molecule consists of two strands, twisted around each other to form what's known as a double helix. Each DNA strand is made up of a chain of 4 different nucleotides, the basic units or letters of DNA. These nucleotides are composed of a sugar called deoxyribose, a phosphate group, and one of 4 nitrogenous bases, adenine, cytosine, guanine, or thymine, abbreviated as A, C, G, and T. The nucleotides on one strand pair with complementary nucleotides on the opposite strand through hydrogen bonds. Specifically, A pairs with T via 2 hydrogen bonds, and C pairs with G through 3 hydrogen bonds. Moreover, DNA strands have a defined orientation. One runs in the 3' to 5' direction, while the other runs from 5' to 3'. Think of it like 2 intertwined snakes coiled together, but heading in opposite directions. All of the proteins in our bodies are encoded by unique arrangements of just these 4 nucleotides. When mistakes happen in our genetic code, diseases can develop. And truthfully, we've always been curious to uncover the secrets hidden within our DNA. Polymerase chain reaction, commonly known as PCR, is a molecular biology technique used to amplify specific segments of DNA. Let's rewind a bit. A single DNA copy is actually a very small amount to work with. To make it easier to study, we generate many, many copies of that DNA, essentially multiplying it to workable amounts. This way, it becomes much simpler to analyze. For instance, if we want to see the DNA, we can apply a method called gel electrophoresis. PCR relies on DNA replication, the natural process our cells use to copy their genetic material before splitting into 2 identical daughter cells. First off, we need a device called a thermal cycler. That's where all the PCR action takes place. Think of it as a cauldron filled with a special solution where genetic wizards add their ingredients. These ingredients include the DNA we want to replicate, an enzyme known as Taq Polymerase, specific primers that attach to the DNA, and a mix of free nucleotides, A, T, C, and G. Toss everything into the thermal cycler, give it a figurative wave of your magic wand, and the process kicks off. So, imagine we have a long, double-stranded DNA molecule, and we're particularly focused on a specific highlighted region. These 2 strands act as the template strands. The first step in PCR is called denaturation, where we raise the temperature to exactly 96 degrees Celsius, nearly as hot as boiling water. This high heat breaks the hydrogen bonds between the 2 DNA strands, causing them to separate. Next comes the annealing step. This is where primers come into play, and for our highlighted region, the primers would be 3'-C, GGATAC-5', 5'-AGGTCAC, 3'. During annealing, we reduce the temperature to about 55 degrees Celsius, which allows the primers to bind to their matching sequences on the single-stranded DNA. The third step is known as extension. And here we aim to create the ideal environment for Taq polymerase to get to work. Taq polymerase is quite an impressive enzyme. It originates from a bacterium called Thermus aquaticus, which thrives in geothermal hot springs. Thanks to this origin, Taq polymerase not only survives the high temperatures of denaturation, but also works optimally at around 72 degrees Celsius. So, in the extension step, we raise the temperature to 72 degrees, and Taq polymerase binds to the primers, picks up free nucleotides, and builds the new DNA strands. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. And that's basically all there is to it. A complete PCR cycle takes around 10 minutes, and by the end, the DNA amount is doubled, since each template strand now has a newly synthesized complementary strand. From here, we simply repeat these 3 steps for several cycles, and with each round, the DNA amount doubles. You start with 2 strands, then get 4, then 8, 16, 32, 64, 128, and so on. This means PCR amplifies DNA exponentially. After about 6 hours and 40 cycles, you could theoretically get 2 to the power of 40, or 1,511,627,776 copies, which is easier to describe as a massive amount of DNA ready for further analysis. There are many PCR variations out there, but let's focus on one that was recently used for detecting a viral infection. For instance, suppose you wanted to diagnose someone with COVID-19. To do this, you would use a nasopharyngeal swab, essentially a long Q-tip with a soft tip, and gently swirl it a few times inside the nostril to collect enough secretions for analysis. Collecting a proper sample is very important, so it's essential to insert the swab deep enough. If the virus isn't picked up, PCR won't be able to detect it. To confirm a COVID-19 case, the target is the virus called Severe Acute Respiratory Syndrome Coronavirus 2, or SARS-CoV-2. Now, SARS-CoV-2 is an RNA virus, not a DNA virus, so in order to detect it, you need to use a specific type of PCR known as reverse transcriptase PCR, or RT-PCR for short. This method works a bit differently because it begins with RNA instead of DNA. If you recall, one key distinction between RNA and DNA is that RNA lacks thymine. In its place, RNA contains uracil. However, RNA is still complementary to DNA. For example, if an RNA strand has the nucleotide sequence 3' AAGU CCAGU 5', then the complementary DNA, or cDNA, strand would be 5-TTCAGGTCA-3. To carry out RT-PCR, a portion of the extracted sample from the nasopharyngeal swab is mixed with a solution that contains an enzyme called reverse transcriptase, along with nucleotides and primers that are specific to a target sequence in SARS-CoV-2. If viral RNA is present, the primers bind to the RNA strand, and reverse transcriptase then builds a complementary DNA-cDNA strand. Once the cDNA is formed, the rest of the RT-PCR process mirrors standard PCR. Taq polymerase is used to amplify the cDNA through the usual denaturation, annealing, and extension phases. After multiple cycles, If SARS-CoV-2 RNA was present in the original sample, you'll end up with amplified DNA that can be detected. All right, let's do a quick recap. Polymerase chain reaction is a method used to greatly amplify DNA, making it easier to examine with other analytical techniques. It involves the use of a thermal cycler, which contains the DNA sample to be amplified, heat-resistant Taq polymerase, primers that determine the specific DNA region to replicate, and free nucleotides of all four types, A, T, C, and G. The process includes three main steps: denaturation, annealing, and extension. With each cycle, PCR doubles the quantity of the specific DNA segment we're targeting. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!